In 1961, a young anatomist named Leonard Havlik made a discovery that would destroy one of humanity's oldest dreams. He was working at the Wistar Institute in Philadelphia, studying how cells grow in laboratory cultures, when he noticed something that contradicted everything his colleagues believed. Cells were supposed to be immortal. That was the scientific dogma of the time. Since 1912, when Alexis Carrel, a Nobel Prize-winning surgeon at the Rockefeller Institute, claimed to have kept chicken heart cells alive and dividing for decades in his laboratory. Scientists believed that cells could reproduce indefinitely. Death, they assumed, was a problem of the organism as a whole, not of individual cells. If you could just provide the right nutrients, the right environment, the right conditions, cells would keep dividing forever. Carrel's immortal chicken heart became famous. It was cited in textbooks. It was referenced in scientific papers. It became one of those facts that everyone knew cells are immortal, and only the body that houses them is mortal. Havlik found otherwise. He was growing human fibroblasts, connective tissue cells taken from fetal tissue in carefully controlled laboratory conditions. The cells divided regularly as expected. They doubled in number, then doubled again, then doubled again. Everything seemed normal for weeks and months. Then, without warning, they stopped. After approximately 50 divisions, the cells simply refused to divide anymore. They didn't die immediately. They entered a strange twilight state that Havlik called senescence, a kind of cellular retirement. The cells were still alive, still metabolizing, still consuming nutrients, but they never divided again. No matter what Havlik did, no matter how carefully he maintained the culture conditions, no matter what growth factors or nutrients he provided, the cells had reached some invisible limit. Havlik was puzzled. He repeated the experiment with different cell lines. He tried different culture techniques. He had colleagues check his methods. The result was always the same. Human cells could divide approximately 40 to 60-50 was a good average. And then they entered senescence and stopped dividing permanently. It was as if the cells were counting, as if they had some internal mechanism keeping track of how many times they'd reproduced. And when they reached the predetermined limit, they quit. This was heresy. Hayek's peers didn't believe him. They assumed he'd made some kind of error. Perhaps he'd contaminated his cultures. Perhaps his technique was flawed. Perhaps he'd somehow introduced a defect that caused the cells to stop dividing prematurely. After all, Carrel's immortal chicken heart cells had been dividing continuously for 50 years. Havlik, a young scientist without Carrel's prestige, must simply be wrong. The scientific establishment pushed back hard. Havlik's papers were rejected by major journals. His findings were dismissed as artifacts. Senior scientists assured everyone that cells were immortal. They'd known this for decades, and this young upstart simply didn't know what he was doing. But Havlik persisted. He conducted more experiments. He documented his methods meticulously. He invited skeptics to visit his laboratory and observe his techniques firsthand. Slowly, other researchers began to replicate his findings. Cells from humans, from mice, from various other organisms all showed the same pattern. Division, 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 and then stop. Meanwhile, something troubling was happening to Carrel's immortal chicken heart. Carrel had died in 1944, but his cell culture was maintained by his successors at the Rockefeller Institute. It kept dividing year after year, seemingly proving that cells could be immortal. But when other laboratories tried to replicate Carrel's results, they couldn't. Their chicken heart cells always died after a limited number of divisions. Only Carrel's original culture seemed to be immortal. Eventually, investigators discovered the truth. Carrel's supposedly immortal cells had been contaminated. Every few days, when technicians added fresh chicken embryo extract to feed the culture, they inadvertently introduced new living cells from the extract. The immortal cell line wasn't immortal at all. It was being constantly replenished with fresh cells, like a river that seems eternal, but is actually made of constantly changing water. Carrel had been fooling himself and the entire scientific community for over 50 years. Havlik was vindicated. His discovery, now called the Havlik limit, became one of the foundational facts of cellular biology. It appears in every textbook. It's taught in every medical school. It fundamentally changed how we understand life and death at the cellular level. Human cells are not immortal. They're born with a countdown timer built into their very DNA. And when that timer runs out, they stop dividing forever. I want you to understand why this matters for the dream of human immortality. You are not a single entity. You are a colony of approximately 37 trillion cells, each one a complex living system in its own right. These cells are constantly dying and being replaced. Your skin cells last about 2 to 3 weeks before they're shed and replaced. Your red blood cells live. about 4 months. The cells lining your gut are replaced every few days. Even the cells in your bones are constantly being broken down and rebuilt. This constant turnover is essential for life. Cells wear out, they accumulate damage. They make mistakes when copying their DNA. Without replacement, your tissues would degrade rapidly. You'd be dead within weeks, killed by the failure of cells that were never renewed. The Halleck limit puts a cap on this renewal. Each cell can only divide so many times. Once a cell has used up its allotted divisions, it can no longer contribute to tissue maintenance. It either dies or enters senescence, a zombie state where it takes up space and even secretes inflammatory chemicals, but never divides again. As you age, more and more of your cells reach this limit. Your body's capacity for self-renewal diminishes. Tissues that were once vibrant with dividing cells become populated by senescent cells that can't contribute to repair. Wounds heal more slowly. Organs function less efficiently. Systems that once maintained themselves now gradually deteriorate. This is aging at the cellular level, and it's built into the fundamental architecture of your biology. But what causes the Halleck limit? What is the counting mechanism that tracks division? And a few dozen base pairs per division doesn't matter, there's plenty of buffer. But as divisions accumulate, the telomeres get progressively shorter. After 50 divisions or so, they become critically short. At this point, the cell faces a crisis. If it continues dividing with critically short telomeres, the ends of its chromosomes might be mistaken for broken DNA. The cell's repair machinery might try to fix the problem by fusing chromosomes together, causing catastrophic genetic damage. Alternatively, essential genes near the chromosome ends might be lost, killing the cell or turning it cancerous. To prevent this disaster, cells have a fail-safe mechanism. When telomeres get too short, the cell triggers a program called replicative senescence. It stops dividing permanently. It's still alive, but it will never reproduce again. This is the molecular basis of the Halleck limit. Telomere shortening is the counting mechanism. Each division shortens the telomeres. When they're short enough, the cell shuts down. Now, you might be thinking, isn't there a way to prevent telomere shortening to rebuild the telomeres after each division so cells can keep dividing forever? There is. Your body has an enzyme called telomerase. Discovered by Carol Giarder and Elizabeth Blackburn in 1984, telomerase can add new repeats to telomeres, rebuilding them and potentially allowing unlimited division in certain cells, stem cells, germ cells, some immune cells. Telomerase is active. These cells can divide far more than 50 times because they keep rebuilding their telomeres. This is essential for life. You need stem cells that can keep dividing to replenish your tissues. You need germ cells that can divide to produce eggs and sperm. So why don't all your cells have active telomeres? Why does your body just keep all its telomeres long and avoid the whole problem of cellular aging? The answer is cancer. Cancer cells are in a sense immortal. They've escaped the normal controls on cell division. They divide and divide, forming tumors that grow without limit. And one of the key ways they achieve this immortality is by reactivating telomerase. About 85%, 90% of human cancers have active telomerase. This allows cancer cells to maintain their telomeres indefinitely dividing forever without hitting the Halleck limit. Telomerase is one of cancer's essential tools. This creates a horrifying trade-off. The same enzyme that could theoretically make your cells immortal also enables cancer to kill you. Your body suppresses telomerase in most cells specifically to prevent cancer. The Halleck limit, the countdown to cellular senescence, is actually a tumor suppression mechanism. You age because your body is protecting you from cancer. The cells that could divide forever are the cells most likely to become malignant. By limiting cell division, your body sacrifices longevity for safety. Some researchers have tried to thread this needle to reactivate telomerase in a controlled way that extends lifespan without causing cancer. The results have been mixed at best. In mice, telomerase activation can extend lifespan, but it also increases cancer risk. The trade-off doesn't go away just because we understand the mechanism. Evolution had hundreds of millions of years to solve this problem. If there were a way to have active telomerase without increased cancer risk, natural selection would have found it. The fact that no complex organism has evolved unlimited telomerase activity suggests that the trade-off is fundamental, not a problem to be solved, but a constraint to be accepted. This brings me to a deeper point about why immortality is impossible. The Halleck limit isn't a bug. It isn't an accident of evolution that we can engineer away. It's a feature, a carefully calibrated system that balances the need for tissue renewal against the risk of cancer Disrupting this balance doesn't lead to immortality. It leads to disease. Your body is full of such trade-offs. Your immune system protects you from infection, but can also attack your own tissues, causing autoimmune disease. Your metabolism produces energy, but also generates free radicals that damage your cells. Your DNA repair mechanisms fix mutations, but can also cause problems if they become overactive or underactive. Aging is the cumulative result of all these trade-offs. You don't age because of one single problem that can be fixed. You age because life is a balancing act among dozens of competing demands. And those balances inevitably shift over time. Leonard Hayek understood this in a 2007 interview. He said, "The reason we age is not because something goes wrong. The reason we age is because something goes right." What he meant was that aging is the natural consequence of biological processes working as they're supposed to work. Cells divide and accumulate damage. Telomeres shorten to prevent cancer. Senescent cells accumulate because the alternative continued division of damaged cells would be worse. Hayek is still alive as I record this in his mid-90s. He has outlived most of his critics, but he hasn't escaped the limit that bears his name. His cells have been counting down for nearly a century, and the countdown is approaching its end. The man who discovered why we age is still aging. The scientist who revealed the cellular clock is still subject to it. Knowledge doesn't grant exemption from the laws it describes. This is the first wall you hit when you try to achieve immortality. Your cells are programmed to stop dividing. The mechanism is telomere shortening. The reason is cancer prevention. The result is aging and death. But this is only the beginning. Behind the Hayek limit, there are other walls. Walls built from physics and thermodynamics and information theory. Walls that no technology can breach. Let me show you what I mean. In 1956, a British scientist named Denham Harman proposed an idea that seemed almost too simple to be true. He suggested that aging was caused by molecules called free radicals, highly reactive chemical species that damage cells from the inside. Harman was working at the Donner Laboratory in Berkeley, California, studying the effects of radiation on living tissue. He knew that radiation damages cells partly by generating free radicals, molecules with unpaired electrons that desperately seek to react with other molecules, stealing electrons and causing chemical havoc. But Harman realized something crucial. You don't need radiation to produce free radicals. Your own metabolism produces them constantly as a byproduct of generating energy. Every cell in your body contains hundreds or thousands of mitochondria, tiny organelles that function as cellular power plants. Mitochondria take the food you eat and the oxygen you breathe and convert them into ATP, the molecular fuel that powers virtually every cellular process. This is aerobic respiration, and it's essential for complex life. But the process isn't perfectly efficient. As electrons flow through the mitochondrial machinery, some of them escape. These escaped electrons react with oxygen to form superoxide radicals, hydrogen peroxide, and other reactive oxygen species. Your mitochondria are constantly leaking these dangerous molecules as a byproduct of keeping you alive. Harman proposed that this constant bombardment of free radicals gradually damages cellular components, proteins, lipids, and most importantly, DNA. Over time, the damage accumulates. Cells become less functional, tissues degrade, the organism ages. This is the free radical theory of aging, and it's been one of the most influential ideas in gerontology for nearly 70 years. The evidence for oxidative damage is overwhelming. As organisms age, they accumulate oxidized proteins, damaged lipids, and mutated DNA. The markers of free radical damage increase with age in virtually every tissue studied. Cells from old animals show more oxidative damage than cells from young animals. The correlation is undeniable. But here's where things get complicated. If free radicals cause aging, then antioxidants should slow it down. Antioxidants are molecules that neutralize free radicals. They donate electrons without becoming reactive themselves, breaking the chain reaction of oxidative damage. Vitamin C, vitamin E, beta-carotene, these are all antioxidants that can neutralize free radicals in test tubes. So researchers tried the obvious experiment. They gave animals antioxidant supplements expecting them to live longer. They didn't. Study after study found that antioxidant supplementation doesn't extend lifespan in laboratory animals. In some cases, it actually shortened lifespan. High doses of vitamin E, for instance, have been associated with increased mortality in human clinical trials. The antioxidant hypothesis, which seems so promising, failed the most basic test. Why? Because the relationship between free radicals and aging is more complicated than Harman imagined. Free radicals aren't just damaging byproducts, they're also signaling molecules. Your cells use reactive oxygen species to communicate, to trigger adaptive responses to activate stress resistance pathways. When you exercise, your muscles produce large amounts of free radicals. This isn't harmful, it's essential. The free radical burst triggers adaptations that make your muscles stronger, your mitochondria more efficient, your antioxidant defenses more robust. Taking antioxidants after exercise actually blunts these beneficial adaptations. Similarly, mild stress, including mild oxidative stress, triggers a response called hormesis. The organism becomes more resilient, better able to handle future challenges. This is why exercise is healthy despite producing free radicals, why fasting can extend lifespan despite being a metabolic stress, why moderate amounts of certain toxins can be beneficial. The free radical theory wasn't exactly wrong. Oxidative damage does accumulate with age and it does contribute to aging, but it was too simple. You can't just neutralize free radicals and expect to live forever because free radicals are woven into the fabric of cellular regulation. This illustrates a broader problem with attempts to achieve immortality. Biological systems are complex. They involve countless interacting components, feedback loops, trade-offs, and redundancies. You can't fix one thing without affecting everything else. Let me give you another example. Protein aggregation. Your cells are full of proteins, molecular machines that perform virtually every cellular function. Proteins must fold into precise three-dimensional shapes to work properly. A misfolded protein is like a crumpled instruction manual. Useless at best, dangerous at worst. Your cells have quality control systems to handle misfolded proteins. Molecular chaperones help proteins fold correctly. Proteo and autophagy systems identify and destroy proteins that can't be salvaged. Under normal conditions, these systems keep misfolded proteins under control. But the systems aren't perfect. Some misfolded proteins escape destruction, some aggregate together forming clumps that resist degradation. Over time, these aggregates accumulate. You've heard of some diseases caused by protein aggregation. Alzheimer's disease involves aggregates of amyloid beta and tau proteins in the brain. Parkinson's disease involves aggregates of alpha-synuclein. Huntington's disease involves aggregates of mutant huntingtin protein. These are all diseases of protein misfolding and aggregation. But aggregation isn't limited to disease states. Even in healthy aging, protein aggregates accumulate throughout the body. Your quality control systems become less efficient with age. Partly because the proteins that make up those systems are themselves subject to damage and misfolding. It's a vicious cycle.